All right, next we will hear from Hubert Zhou, who is from Daiichi Sankyo, working on a clinical trial in phase 2. Wilco, you're up. Hubert, you're up next.
All right. Can you see my slide?
Just about. I still see it starting sharing screen, but that could be, yep, here we go.
All right. Well, thank you very much, Sharon. Let me go ahead and in this segment, give an update on the TNAP inhibitor for PXE, and I will speak from this as a DS-1211 as an investigational drug, and I'm, as Sharon said earlier, I'm from Daiichi Sankyo, so I will provide this from a drug development perspective from pharmaceutical company. So you have seen this slide many times, and the main thing, let me hang on a second, let me just do pointer, all right? And really this, as a reminder, this cartoon we all have seen many times, and this is just a reminder, this mechanism, but as being talked about during this meeting a few times, the central part is really this pyrophosphate. And in people with PXE, we recognize that it's reduced. It's about 40 to 50%. And we believe that this relates to the ectopic calcification because the pyrophosphate is a very potent endogenous inhibitor of calcification. And itself is cleaved or inactivated by this enzyme called TNAP, or tissue non-specific alkaline phosphatase. And the question is, if we were to inhibit it, will we increase the pyrophosphate level and subsequently reduce the calcification? So in developing such a compound, a TNAP inhibitor, there are many questions that we need to ask, right? And one, if we can do that in the test tube, would this compound inhibit a TNAP in the animal? And if so, would it increase the pyrophosphate level in the animal? And subsequently, we need to really know, would it reduce the ectopic calcification? And more importantly, we need really to understand then what would happen in human. After all, human are not these mice or monkeys. So this is the next few slides. I'll just show the results that we test this compound DS-1211 in the animal. On the left here is showing that this compound is very potent, and the graph here is showing the increasing concentration and the level of inhibition. As we increase the concentration to higher and higher, is simply more inhibition occurred. Great. And this occurred in the mice, in the monkey, and also in the human cells. This is in the test tube. On the right-hand graph, show that this particular inhibitor is very specific for TNAP, a tissue non-specific alkaline phosphatase, as compared to other alkaline phosphatases. The difference in its potency, if you would, is about 1,000-fold. So it's specific for this particular enzyme. And you say, great, then what does it do? Does it really, in the live animal, like mice, would it really show inhibition is beyond the test tube? And the answer is indeed yes. On the left here, we show that I'm showing you the same kind of set, and this is the time after administration, and this is the plasma alkaline phosphatase level by measuring the activity. And what you see is the activity goes down after you dose the mice with the TNAP inhibitor. Would it increase the pyrophosphate level? And the answer is also yes. After you administer the compound, the pyrophosphate level goes about 2-fold higher in the about 1 to 4 hours after dosing. And you say, it's great, all right, you can show that, but does it really reduce the calcification itself? And as you can, you have heard from many other presentations that in the mice, we're using the calcification in the vivisa, in the whisker, as a measure. And you also have heard in the previous talks about animal models, including the ABCC6 knockout mice, and also another strain of mice called KKHIJ mice. And that's indeed what we tested. And we show in here that in this particular PXE mice model, KKHIJ, you can see the reduction in the alkaline phosphatase is inhibited. The higher concentration, the more inhibition there is, and the calcification is reduced comparing to that of control. Same is true in another mice model, PXE, ABCC6 knockout mice. Alkaline phosphatase is inhibited, and the calcium in the revisa is reduced. And you say, it's great, all right? So in what I've shown you here is with this particular TNAP inhibitor would inhibit TNAP in the animal, would increase the pyrophosphate in the animal, would reduce the calcification. How about in human? And we really, and you know, human is, again, human are not mice, and we're putting this new chemical entity into human, and we need to be very deliberate on the steps that we take. We need to be very careful, and we need to be very methodical. So we first want to test the TNAP inhibitor in human, in healthy people, and ask the same question of would it safely inhibit TNAP, and would it safely raise the pyrophosphate level? And then we're going to test in people with PXE. So let me show you in the next few slides the work that we have done, the study we have completed in healthy individuals. So that's called the phase 1 program. And the objective here is, one, would the compound get into the body? Would it be safe? And would it have the desired effect? We tested in what's called a single, and then the repeat dose studies. And then we also tested formulation, a tablet formulation to be specific, and also test the food effect. So the first single dose study, again, you know, we need to be very careful. We need to be very methodical. We think based on the animal results, about 30 milligrams is where we're going to see a good results in terms of inhibiting the TNAP and raising the pyrophosphate. So we purposely started a very low dose, 3 milligrams. We finished that cohort. We tested. We're satisfied with the result in terms of safety and the effect. And then we move on to the next cohort, 10 milligrams, and so on, gingerly stepping up. In this study, we actually went all the way to 3,000 milligrams. After that, we take the results and we use the dose between 10 and 300 milligrams and move on to the 10-day repeat dose study. And again, it's a stepwise, careful, methodical way of escalating the dose to the next cohort. We finished that. We also then tested versus the powder formulation used in the earlier study, in the single and repeat dose study. We tested the formulation, a tablet formulation that will be used in the outpatient study. So I'll just show you the result from the repeat dose study. This is a way to assess the safety, and this is a participants reporting of the adverse reactions. And what's listed here is in each column, at each dose cohort, 10 milligrams all the way to 300 milligrams a day, either single dose or twice a day dosing. And what you can see is most of these adverse reactions reported are typical of what one might encounter in the daily living. More importantly, you don't, as the dose increases, you don't see increase in the adverse reaction type or frequency, suggesting that the dosing itself, the compound itself, is relatively safe and well tolerated. Do we see a pyrophosphate increase? I'm pretty sure you'll be asking that question as well. And the answer is indeed. And just like what we have seen in the PXE mice model, we see after dosing, and this is 10 days after the last dose, after that dosing on day 10, you see from 10 to 30 to 100 milligram, you see the increases in pyrophosphate right after dosing. Right. So that's great. We say animal checked, safely inhibit TNAP and raise the pyrophosphate up to 10 days, check. What's next? Well, we need to move on and to test in people with PXE. And that's it is what we are doing right now. Let me show you that. And this is the study. This is the phase 2 study in people with PXE. And in this case, it's a randomized double-blind placebo-control. All that really means is we're going to be comparing it and to an inner agent, and without biasing the investigator or the study participant. And so they will not know what they will be receiving, and we'll be analyzing the result at the end of the study. It's a 12-week study. We're testing 3 active doses, and we'll be assessing primarily safety, because that's obviously the most important. We'll be assessing the concentration, how well get into the body. We're assessing the effect, including the desired effect, and also monitoring for the potential safety factors. So with that, we know this is not going to be the end, right? Because raising the pyrophosphate is not enough. And as you have heard many of the talks in this conference, we'll be very interested in whether the compound like this would safely reduce the calcification, that's undesirable calcification, in people with PXE, whether that's in the blood, in the eye, in the skin. And that's not, certainly it's not very easy to measure. Wilco had mentioned about and described the attenuation trial using the CT calcium. That can be done. We also need to be monitoring, like Christina had mentioned, perhaps in the Brooks membrane and in the eye, and we can test that in the skin through biopsy if necessary. We simply want, and these, because calcification takes time, this will be 1 to 2 years and maybe even for longer follow-up. So it will take time, and the dosing will be much beyond the 12 weeks. Would that be enough? Well, no, because you and also have described and discussed so much about your concerns, right? You're not concerned specifically just on the calcification that we can measure by various kind of tests, but it's really what's important is how will this help you to be to function better, to feel better, whether it's in your eye, whether it's in walking without claudication, or would it really improve even the skin? We don't know. But these, because it's such a slowly progressive disease, it's going to take a long time. But that is the end goal. It's really to understand and test and ask these questions. Will compound like this be safely improve the signs and symptoms in people with PXE? And that will take a long time, but we need to be patient because that ultimately is the most important part. And there are a lot of different endpoints. We talk about biomarker or clinical study endpoint. That's something that we're focusing on and working with the entire research community. So with that, thank you very much. And particularly many thanks to the people with PXE and the family and many of the collaborators that are listed here. But lastly, I want to mention really in fond memory of Dr. Juni Yurita. He's been such a great advisor, collaborator, and a teacher, and really a friend. I miss him. I miss him greatly. So thank you.
Yes. Thank you, Hubert. I should also say that we're working very closely with Daiichi Sankyo. They've been a company that's recognized my expertise as well as PXE International's contributions. And so I'm an investigator in the trial, which is really makes it easy to be involved in the recruitment and so forth. So we're helping with all of that. So Hubert, a question: if successful, then what happens next?
Successful by that, I assume you mean this ongoing trial. Then we'll be obviously moving on to planning, and we're planning now for the next study. And again, the next step will be really understanding and really demonstrating that this compound can reduce the calcification in people with PXE.
Okay, other questions? We're still recruiting for those who are interested, especially looking in the Philadelphia, New York, eastern seaboard of the US area.
Maybe just make one more point there, Sharon. Just, speaking of safety, and I want to emphasize this because why I was saying that we need to be very deliberate, we need to be very methodical. It's really, we need to really with an eyes to safety. After all, PXE is not acute life-threatening in people with PXE, and we need to, and safety by that I mean side effect. And we don't want even bad side effect in 1 in 1,000 people. And that is not just taking for 10 days. It needs to be taking for long term. So we and the FDA and other regulatory agency and ethics committee obviously will be doing the utmost to safeguard the participants' safety. So make a pitch there.
Great. The other questions? Okay, thanks, Hubert. And so I think you all can see that once in a while people say, what are you doing with our money we're donating? And you can see that we're really supporting a great deal of research in different areas. Certainly when a company gets involved, the company pays for most of the research, in fact, all of it in this regard. But all of the time that we've spent 28 years leading up to this, and certainly even before PXE International, has been critical to getting us to this place, because it's really the combined efforts of lots of activity that brings us to a place where we start to understand. You also can see that it's a very collaborative community in the sense that we're all rowing in the same direction, and there is no competition in the sense of discoveries are shared with one another. We had a researcher meeting in the fall. We do that once a year, either in the US or Budapest, where Andras Vardy sponsors that. And the researchers all get together and share cutting edge research that has not yet been published. And so we're really pushing as fast as we can to get something ready for development.